IB Bio, Topic 11, Part 12, Sexual Reproduction D, skims through the development of the embryo into the fetus, then bears down on the details surrounding birth. The essential idea is sexual reproduction involves the development and fusion of haploid gametes. Here is the outline of all the available movies for Topic 11, Animal Physiology. Use this outline to find the movie you need for review. This movie is focused here. Remember that mammals utilize internal fertilization as an evolutionary adaptation to living on land, a dry environment. And as a result of employing internal fertilization, mammals provide high protection to the development of their young, not to mention giving birth to just a few young. When mating couples give birth to low numbers of offspring, it requires a high investment of parental energy in the survival of their mammalian offspring. In this slide, we can see the relative growth of the fetus over 40 weeks, 9 months. In the first trimester, the following events occur. Implantation, week 1. There are recognizable brain regions by week 4. The circulatory system is functioning, the heart beats by week 4. The respiratory system and the digestive system are functioning by week 6. The limbs are distinguishable by week 6, and the sex can be determined by week 8. Here are photographs of the fetus at 4 weeks and 8 weeks of the first trimester. Here are photographs of the fetus at 12 weeks and 16 weeks, the start of the second trimester. At the start of the second trimester, the corpus luteum deteriorates, and the placenta picks up secretion of progesterone and estrogen to maintain the uterine lining. In the second trimester, the following events occur. The fetus grows in length, body systems approach functional. There's a breathing behavior that can be observed. It's fluid in and out of the mouth. It's not actually respiration in the sense of bringing in gases for gas exchange. The fetus responds to stimulus in the second trimester and begins to kick in the 16th week. Here are photographs of the fetus at 20 weeks of the second trimester. In the third trimester, the following events occur. Organ systems become fully functional. The fetus grows to 35 centimeters, 1 kilogram. Ossification occurs. This is development of the skeletal system. The brain develops, and antibodies from the mother provide passive immunity to the fetus. Here are photographs of the fetus at 28 and 36 weeks of the third trimester. In this diagram, we can see the fetus at 8 months, 1 month prior to birth. The fetus has turned upside down in preparation for delivery, head first. This is a good image of the infant late in pregnancy. You can see the amniotic sac and the fluid within. Both of these structures protect the baby through the gestation period. Also notice the placenta. At birth, muscles of the uterus will contract, known as labor, to push the infant out through the cervix and vagina. In these images, you can see the development of the fetus in the uterus over the first 20 weeks of gestation. In these images, you can see the development of the fetus in the uterus from week 24 to week 40. Here's an image of the infant at 8 months. Notice the position of the body relative to the cervix. At birth, the cervix will dilate to allow the baby to emerge. Also notice the placenta. Here is an image of birth. The uterine walls are contracting under the influence of the hormone oxytocin. The contractions, stimulated by oxytocin, help push the infant out of the uterus. Oxytocin is secreted from the posterior pituitary. Here is the first IB syllabus statement of the movie, a statement addressing the relative size of mammals with the length of gestation. The average 38-week pregnancy in humans can be positioned on a graph showing the correlation between animal size and the development of the young at birth for other mammals. This graph displays the relationship between adult body mass and gestation period in a range of mammals. You can see the generally positive correlation between adult size and gestation period. However, there are mammals with the same gestation period but widely varying body masses, and this is on an order of greater than 10 to the third. The black data points are altricial mammals, mammals that give birth to very immature young, like humans. The white data points are precocial mammals, mammals that give birth to young ready to defend themselves at birth. Altricial mammals tend to have longer gestation periods than precocial mammals. Wildebeest give birth to highly developed young ready to run within a few minutes of birth. Wildebeest are precocial. Humans give birth to very underdeveloped young who take at least a year to walk. Humans are altricial. Here is the next IB syllabus statement of the movie, a statement surrounding the details of birth. Outline the process of birth and its hormonal control, including changes in progesterone and oxytocin levels, and positive feedback. Here is an image of birthing. The uterine walls are contracting under the influence of the hormone oxytocin. The contractions, stimulated by oxytocin, help push the infant out of the uterus. Oxytocin is secreted from the posterior pituitary. Now, birthing does have various stages. There's labor, the contractions of the uterus, delivery, and the afterbirth. The afterbirth, as you can see here, involves the removal of the umbilical cord and the placenta. I've shown you this graph in the previous movie, Part 11, Reproduction C, when the focus was on HCG levels during the first 3 months of pregnancy. I'm showing you this graph again for the purposes of looking at the changing levels of progesterone and estrogen. Notice their blood levels increase through the 9 months due to the hormone secretion from the placenta. But the decline of progesterone and to some extent estrogen at the end of the gestation period is the stimulus for birth. Take note of this. Here is another graph showing blood concentrations over the gestation period of HCG, progesterone, and estrogen. Notice the dramatic decline of blood progesterone at the end of the gestation period. The decline of progesterone triggers labor, the first stage of birthing. The decline of progesterone releases the inhibition on oxytocin release from the posterior pituitary. With increased levels of oxytocin in the blood, uterine contractions begin. Labor. So here is an outline of the stages of birthing. First, hormonal control. Estrogen from the placenta sensitizes the uterus to oxytocin. In other words, receptors to oxytocin become more numerous on the uterus. Progesterone levels decrease. I mentioned this in the last couple of slides. And oxytocin being secreted from the posterior pituitary rises, targeting the uterus. Then labor, uterine contractions begin, dilation of the cervix, amniotic sac breaks. And then delivery. The infant emerges, head and body through the vagina. The umbilical cord is cut. The afterbirth, which is the placenta, is removed. Here's the afterbirth. The placenta separates from the uterine wall. Progesterone levels decline further. Prolactin secretion occurs, so milk production is stimulated. With the decline of progesterone at the end of the gestation period, oxytocin is released from the posterior pituitary. Oxytocin targets the uterus to stimulate muscle contractions. Contractions of the muscle sends nervous signals back to the posterior pituitary, back to the hypothalamus, so that there's more release of oxytocin. This is positive feedback. More contractions, more neural signals to the hypothalamus and the posterior pituitary, more oxytocin. Thus, the urgency around giving birth. Take note of this. positive feedback with oxytocin and uterine contractions. This is labor. Estrogen further enhances the positive feedback by increasing receptors to oxytocin on the uterine wall. Again, neural signals from the uterus to the hypothalamus results in the release of more oxytocin. More oxytocin, more muscular contractions, more oxytocin, more contractions. Delivery through a positive feedback process between uterine contractions and oxytocin release. One of estrogen's main roles leading up to birth is to sensitize the uterus to oxytocin. Estrogen stimulates an increase in oxytocin receptors on the muscle cells of the uterus. This enhances the positive feedback effect of oxytocin and uterine contractions. Here is one more slide to emphasize the positive feedback surrounding oxytocin and uterine contractions during labor. Study this slide on your own. One more slide. Progesterone levels fall, oxytocin levels rise, uterine contractions increase, oxytocin levels rise, uterine contractions increase, oxytocin levels rise. This is labor. This image also notes the role of estrogen in sensitizing the uterus to oxytocin. There is also a reference to prostaglandins here secreted by the placenta. Do not worry about this detail. At this point, this IB syllabus statement should be under control. It's a decline in progesterone and to some degree estrogen that triggers the events surrounding birth. Uterine contractions, delivery seen here, and then the afterbirth. Here is a photograph of the amniotic sac and placenta after birth. This is known as the afterbirth. To help make connections to earlier parts of this course, I've come back to a small topic that I've presented earlier, in fact, last year. This is a small but easily lost concept. Karyotyping is a technique used to examine fetal chromosomes to determine if the infant is healthy or if the infant suffers from a chromosomal error such as Down syndrome, trisomy 21. Here is the relevant IB syllabus statement. State that karyotyping is performed using cells collected by chorionic villus sampling or amniocentesis for prenatal diagnosis of chromosome abnormalities. Karyotyping starts with the removal of fetal cells using two possible techniques. The first technique is amniocentesis. Fetal cells are withdrawn from the amniotic fluid seen here. The cells are cultured, prompted into mitosis, where the chromosomes become visible. A cell squash is made, and a technician lines up the homologous pairs of chromosomes. Here is an example of a karyogram showing trisomy 21 Down syndrome. The second technique is called chorionic villus sampling. Instead of penetrating the amniotic sac with a needle, a catheter is inserted into the vagina and a sample of the placenta, specifically the chorion, is taken. The cells are cultured into mitosis and a technician arranges the homologous pairs. Chorionic villus sampling has less risk of miscarriage and can be performed earlier with faster results. And that brings us to the end of Topic 11, Animal Physiology, Part 12, Sexual Reproduction, D.